comprehensive identification of microbial pathogens through microscopy, molecular, immunological, and radiological technologies forms the basis for accurate and effective diagnosis of infections, as the precise identification and comprehensive characterization of pathogens like bacteria, viruses, fungi, and parasites provide the foundation for microbiology, clinical diagnosis, and public health. Given that these pathogens are diverse in structure, genetics, and metabolism, a multifaceted diagnostic strategy is essential where modern microbiology integrates microscopy, molecular, biochemical, immune, and radiological techniques to ensure accurate infection, identification, characterization, and clinical management. Bacterial identification relies on morphologic, metabolic, and genetic profiling, where microscopy utilizes gram staining to classify bacteria into gram-positive purple or gram-negative pink, based on cell wall structure, and acid-fast staining, like Zeal-Nielsen identifies Mycobacterium, while morphological analysis reveals size, shape, arrangement, and motility. Additionally, culture and biochemical tests using differential media like McConkey and mannitol salt agar, and enzyme tests such as oxidase and coagulase provide phenotypic signatures for species-level identification. Molecular techniques like PCR, qPCR, and NGs enable the identification of species, genes, virulence factors, and antimicrobial resistance determinants. Immunological methods like ELISA and lateral flow provide rapid detection of bacterial antigens or host antibodies in infections such as H. pylori or Salmonella, and radiological imaging like chest X-rays and CT scans show infection-related structural changes like TB lesions or abscesses. Historically and socially, Pasteur and Koch's culture methods played a central role in establishing the germ theory and identification principles, while in the modern era, rapid molecular diagnosis is gaining increasing importance in resource-limited settings. On the other hand, viruses require sensitive and often indirect diagnostic techniques due to their small size and intracellular nature, where electron microscopy reveals viral morphology and ultrastructure, especially for viruses that cannot be cultured. Molecular diagnosis via RT-PCR and qPCR enables sensitive detection of viral RNA or DNA and monitoring of viral load, as seen with HIV and SARS-CoV-2. Immunological methods like immunofluorescence and Western blot aid in detecting viral antigens or host antibodies in cases like hepatitis B or HIV, and radiological imaging via MRI and CT identifies organ complications of viral infections such as viral encephalitis or liver disease. The discovery of viruses in the early 20th century revolutionized infection diagnosis, making rapid and sensitive identification techniques clinically and socially indispensable, particularly during pandemics.